Assalamu alaikum. Acoustic startle response test has been taken from an article timing of the acoustic startle response in mice, habituation and dishabituation as a function of the interstimulus interval, taken from the journal named International Journal of Comparative Psychology, which is published by the International Society for Comparative Psychology in 2001. And the hypothesis of this test is that the standard acoustic startle response paradigm contains the elements of interval timing. The interval timing may be the standard feature of this habituation paradigm, as it serves as an anticipate the time of the occurrence of the subsequent stimulus and to prepare the startle response and provide a computational dimension lacking in a habituation process per second. Now let me tell you what is acoustic startle response. It is basically a protective behavioral reaction consisting of a muscle contractions of the eyelid, neck, and the extremities that is elicited by the sudden loud acoustic stimuli. The central element is the caudal pontine reticular nucleus, which receives the direct input from the auditory system and directly projects to the spinal and the cranial motor neurons. There are many modulatory influences which affect the primary startle circuit at the level of the PNC. There are some examples of the modulations leading to a decrease of the ASR. These examples are habituation and propulsive inhibition. What is habituation? Habituation is the decrease of the acoustic startle response during a repetitive stimulation. It is assumed to be an intrinsic process occurring within the primary startle pathway. And propulsive inhibition, it is a decrease of the startle amplitude by a propulsive presented shortly before the startle stimulus that does not elicit a startle response itself. The propulsive the propulse activates an inhibiting loop that suppresses the processing of the succeeding sensory inputs. There are some examples of the modulations leading to an increase of the ASR, which is sensitization and a fear conditioning. Sensitization is an enhancement of the ASR following a strong stimulus such as foot shocks or a strong startle stimuli, whereas fear potentiated startle paradigm or a fear condition in which the animals are trained to associate a neutral stimulus with an aversive stimulus. After few pairings, the conditioned stimulus induces a state of fear that can be measured by a potentiation of the ASR. Foot shock sensitization and a fear conditioning are mediated by a direct and indirect projections from the amygdala to the PNC. This broad spectrum of the modulations conclude that methodological advantage that all the experimental procedures can be carried out automatically and by a computer, and we can make ASR a valuable tool for the study of general principles of the plasticity of the sensory motor information processing. So the goal and the aim of this test or an article is to disentangle the habituation of the ASR from the interval time. And what is interval timing? It is basically the ability to estimate the durations in the seconds to minute range. But to achieve this goal, we have to first mm, perform some experimental settings, like we have to give an animal an auditory and a visual signals time to time and receive the food when they make a correct response after a signal duration has passed. After 20 to 30 daily training sessions, the animal's mean response rate gradually increases towards the time of the reinforcement and then declines afterward in a fairly symmetrical fashion. So we will obtain a peak interval procedure, and from this procedure, and we will find out from this study that the timing in various species, including rats, mice, and starling fishes, and we can determine different peak intervals through this study interval timings. Now come to the method. In this test, we have to take 30 subjects, means a total 30 mice served as subjects, and 12 of these mice were male, which are of wild type and 8 to 12 weeks of age, whereas remaining 18 mice were male, black, In color, C57 mice, which is also 8 to 12 weeks of age. These mice were housed in a group of 3-4, and food and water are also available along with the libitum in a home cage, and the vivarium was kept on 12-12 hour light and dark cycle with the lights on at 9th hour. Behavioral testing procedures took place during the light phase of the animal's light dark cycle. Apparatus. The acoustic startle response was measured using four identical startle chambers and associated software controlled by an IBM compatible PC. For the measurement of the ASR, mice were placed in a round acrylic animal holder. In order to determine the appropriate stimulus intensities to use for an experiment with different strains of mice, a preliminary study was conducted prior to the current experiment. 8 mice were placed individually in an acrylic cylinder, and after a 10-minute acclimation period with exposure to a 65 decibel background white noise, 40 millisecond pulse of a white noise at 5 different tone intensities, which are 80 decibel, 90, 100, 110, and 120 decibel, were presented every 60 seconds. Each of the five stimulus intensities was presented once in a randomized order in a block of 5 trials. 10 of these blocks were presented for a total of 50 trials in a session. Based on these input-output functions, an optimum habituation intensity for a white noise stimulus was defined as a midpoint between the minimum startle and a maximum startle response, and which is determined by a standard regression algorithm. A mean optimum intensity value of 100 decibel was obtained from these preliminary studies, and there were no significant strain difference obtained in this measure for the mice used in our studies, which is the value obtained is p is greater than 0.05. On the basis of the preliminary study, we will perform further experiment. Mice were placed in an acrylic cylinder located in a sound, attenuated, dark, and ventilated startle chamber for a 10-minute acclimation period with exposure to a 65 decibel background noise which continued throughout the session. Mice readily adapted to a being placed in a holder and no strain difference was observed. Now, an acoustic startle stimulus consisting of a 40 millisecond pulse of white noise presented at 100 decibel could also be presented. Using random assignment, mice from each genotype were evenly divided in two groups. In group 1, the mice were presented with a continuous sequence of 150 presentations of the startle stimulus, and the first 100 startle stimuli were presented with a 10-second interstimulus interval, immediately followed by 50 trials with 5-second interstimulus interval, and the group designated as 10 seconds to 5-second ISI group, and the mice in group 2 were also presented with a continuous sequence of 150 starter stimuli. The first 100 starter stimuli were presented with a 10-second ISI, then immediately followed by 50 trials with a 15-second ISI. So, ISI group is designated from 10 seconds to 15-second ISI. The latency to a startle is defined by the MAD-ASR Pro software as a time between the onset of the startle stimulus and the onset of the startle response, which is called as a latency to the startle. To the result we obtained from the experiment, as we see this graph, this is a graph of the mean percent acoustic startle response amplitude as a function of the blocks of 10 trials, and these are the groups of the mouse. This is white square shows group 1 mouse, which is 10:5 interstimulus interval group 1, and this black circle shows a group 2 mouse, which is 10:15 ISI, and This vertical line indicates a change in the interstimulus interval, and the data for the two mouse types we show that they are collapsed between, and there was no significant difference in the performance. This is a graph of a person maximum ASR amplitude with a change in interstimulus intervals for the last three blocks of trials. This is a graph which shows the acoustic startle response, maximum acoustic startle response obtained um, by changing in this interstimulus interval condition by the two groups of the mice, which is designated as the group 1, that is 10 seconds to the 5 second, and group 2 10 seconds to the 15 second. But we see that the data collected through this graph is not significant, as we see that there is no significant difference obtained in the performance, and the data for the two mouse types, they are basically collapsed. This is a baseline and this is a test. Through the experiment, we conclude that after a shift in a baseline ISI, we observe the rate sensitivity property of a habituation, that is, increased level of habituation with a shorter interstimulus intervals and decreased level of habituation with a longer stimulus intervals. In addition, immediately after the ISI change, the startle response increased dramatically but quickly rehabituated to the level appropriate to the new interstimulus interval. These results suggest a dissociation of the anticipatory timing and the rate sensitivity mechanism of a habituation. And interval timing interpretations of these results would predict that habituation would be slowed if one were to use a variable interstimulus interval, thereby interfering with the anticipatory timing of the stimulus. In addition, temporal generalization of the ASR appears sufficiently sensitive to serve as a rapid screening assay for interval timing deficit in transgenic and knockout mice. A classic finding in this literature is that habituation occurs faster with shorter ISIs but shows greater maintenance with the longer ISIs. This phenomenon is referred to as a rate sensitivity property of habituation, and it has been demonstrated using a variety of stimuli, that is, pure tones, visual cues, or a tactile stimuli in rats and pigeons. Here we show that this hallmark or habituation can be observed in mice using a white noise as a startle stimulus, and we show that there is evidence for the anticipation of the startle stimulus as predicted by scalar expectancy theory, that a memory of the interval separating individual startle stimulus was revealed by showing increase in the ASR amplitude immediately after a change in the ISI. And consequently, our experiment suggests that there are at least two types of learning involved in the task. One is habituation to the stimulus characteristics of the white noise and its relationship to the background, and the another is the anticipation of the time of the occurrence of the startle stimulus, referred to here as interval timing. Thank you.